But maybe
So I'm saying in endocytosis, we are talking about bringing into the cell. So when you have bigger particles, in this case, we are not talking about the movement of one particular molecule, rather a movement of a number of molecules together, transported in bulk. Endocytosis can either be phagocytosis, which we explained earlier, where we have a bigger molecules or particles like bacteria or virus taken in by an immune cell like phagocyte. We also have phagocytosis. This is usually the movement of a large molecule liquids inside the cell. Or we have a receptor-mediated endocytosis. A receptor-mediated endocytosis, we talked about lysosome last time, and I think I explained how lysosomes has been used in medicine for targeted therapy. When you have a particular drug, for example, and you want that drug after taking in to go directly only to some specific cells, for example, a cancer cells. You know, normal conventional drugs, when you take a drug, you take them, either it's through IV, IM, oral, circulation.
No.
They go into the systemic circulation. When they go into the systemic circulation, they do not differentiate which cells they go in or which cells not go in. They basically absorb, taken up by almost all the cells. In this situation, you want specific cells to take up your drugs. Now you mobilize your drugs into the liposome, as I explained earlier, and you decide to determine some certain proteins on the surface of your membrane of the liposome, so that this protein will only specifically determine some antigens on the surface of these cancer cells. Yes, if you are doing that, you must have an idea about the cancer cells. You must have an idea about some antigens that are presented on the surface of these cancer cells. So you target those antigens. So you present or you design your liposome with this protein on the surface of your liposome, so that when they get into the systemic circulation, they can only attach themselves to these specific cancer cells that have this antigen on them. So attaching on that lead to the uptake of these substances inside the cell. This particular process is what is called receptor-mediated endocytosis. We also have an exocytosis, which we explained earlier, when you have a remnant of digestion from lysosome. You can have a lysosome. A lysosome pinches the cell membrane, and then you have the membrane of the lysosome will fuse with the membrane of the cell, so that the content of the lysosome now be emptied out of the cell. This is the classical of exocytosis. So I have an example here of phagocytosis, which I earlier explained. You have a larger particle, it can be a virus, it can be a bacteria, anything harmful where this immune cell identify as a harmful substances, they can now engulf, form a membrane, and this membrane bound particle, either into bacteria, can now fuse with lysosome for it to be broken down. You have pinocytosis, where you have molecules, fluid, taking into the cell in form of a particle, and you have receptor-mediated endocytosis. And also classical of exocytosis, which I mentioned with regard to lysosome. So here we also have some questions. I will also include these questions to the quiz, so you can try and answer these questions because they will help you. The next thing we talked about is study session 4, Introduction to Nucleic Acid Biochemistry. Here we talked about the chemistry of nucleic acid, the functions of nucleic acid, the component of nucleic acids, and we also know what are nucleotide and nucleoside shortly. So have to pay attention. I hope you are following me.
Yes, sir, we're following.
Yes, sir, we are following.
Good.
Following.
So what are nucleic acids? Nucleic acids, they are organic molecules. Just as I mentioned carbohydrates, lipid, protein earlier, we also have a nucleic acid. And this nucleic acid also, like I earlier explained, excuse me, Kevin. Very good.
Can you talk again?
So, as I was saying, these molecules, just like other organic molecules, they contain or they are made out of carbon, hydrogen, oxygen, nitrogen, and phosphorus. Because we are going to talk about the chemistry of these molecules, of course, you will see how these atoms made up of these nucleic acids. They are first discovered in the nucleus of a cell, and they are found to be a kind of repository and transmitters of genetic information. They are a polymer of nucleotides, meaning they are made up of nucleotides, polynucleotides, linked together by what we call 5 and 3' phosphodiester bond or phosphate bridges, which we are going to see. What is this phosphate bridges? Or what type of bond is this phosphodiester bond? So chemically, we have the nucleic acid divided into two. We have the deoxyribonucleic acid and we have a ribonucleic acid. When you listen attentively, you are going to see the difference between the deoxyribonucleic acid and the ribonucleic acid. When we go back to historically, we'll know that this DNA was first discovered in 1869 by a young Swiss researcher, that there is an acidic substance present in the nucleus, and he called it nucleic. Later on, 1945, it was discovered that, yes, of course, this DNA contained a genetic information by a group of researchers, Avery MacLeod and Marker. In 1953, this is a well-known work of James Watson and Francis Crick, where they describe a simple but a very famous double helical structure of the DNA. The DNA exists in form of double helical structure. Excuse me. I'm having a class tonight. Yes, one of them, yeah. Later on, we have the work of Rosalind Franklin and Maurice Wilkin using study X-ray. They use an X-ray diffraction where they study the DNA fibers, and their findings suggest that these DNA fibers are made up of the helical. They are arranged in a helical conformation. And we also have a work of Arwin Shagaf and Lina Pauling that all contributed in the present knowledge of what we know about nucleic and DNA structure. These are the basis of some of the work of people, and we can see also an X-ray photograph from the work of Rosalind Franklin and Maurice Wilkins. Now, this nucleic acid, what is it made up of? What is it made up of? The nucleic acid is made up of 3 major components. We have nitrogenous base, we have a pentose sugar, we have a phosphate group. 3 major things. So a nucleic acid is made up of a nitrogenous base, a pentose sugar, and a phosphate group. We will see how this nucleic acid comprise of these things. First of all, remember, we said the nucleic acid is a polymer. A polymer of what? A polymer of a nucleotide. A polymer of a nucleotide. So you have polymer of nucleotide. When we say we have a polymer of a nucleotide or polymer of something, meaning that thing attached to another thing of the same kind, also attached to another thing of the same kind, also attached to another thing of the same kind, until you have many of them of similar kind or same kind attached to each other through a particular bond. We call it a polymer. So this is a polymer of nucleotide, meaning nucleotide react with another nucleotide, react with another nucleotide, react with another nucleotide, until you have a very long chain of nucleotide called polynucleotide. And this polynucleotide is what we call the nucleic acid. I think there is a part where we'll discuss about, we continue the discussion from this polynucleotide. How is this polynucleotide even found inside the cell? And then we'll talk about the helical structure, we'll talk about the packaging, the formation of the chromosome, because what we know when we talked about the DNA is that it is found in the nucleus, and inside the nucleus, what we know, it is found in form of chromosome. How this polynucleotide were now packed to form a chromosome. How is the packing formed? We'll discuss about that in the subsequent classes. Class, are we together?
Yes, sir.
Good. So, as I'm saying, this nucleotide as a monomer that is being linked to form a polynucleotide. Each nucleotide is made up of these three things I mentioned: the nitrogenous base, the pentose sugar, and the phosphate group. The nitrogenous base, they are of two types. Or they have two parent molecules. They have purine, which is a parent molecule, and it have sons, and a pyrimidine, which is also another parent molecule, and it also have sons, or it have children. We have a pentose sugar. This sugar, just like any other sugar, you know, even though at this stage, I don't think you know any sugar. Do you know sugar? Do you know sugar?
Yes, sir.
What is the sugar? Can someone tell me one sugar? Yes? Esther. Salome. Joy. Abigail.
So I think we have a simple sugar and a complex sugar. I don't know if I'm right.
Yes. So tell me one simple sugar.
Simple sugar, we have maltose, sucrase, and I don't know.
Okay, you said it and you're saying you don't know. Anyway, you're right. So shortly, I think in the next class, we'll talk about carbohydrate, and there we'll talk about the sugars and their different types. Why I asked this question is just to bring to your notice that the word sugar is not the normal sugar we used to take in the tea. That sugar, we call it a sucrose. And that sugar is made up of two sugars. It's made up of two sugars. It is called sucrose, but it contains two sugars, which is glucose and fructose. Generally, sugars, I will just give you a background of what sugars are so that for you to understand this concept before we discuss about it properly in the next. So sugars are classified based on the number of carbon atoms. Starting with the 3 carbon atoms, there are sugars that contain only 3 carbon atoms, and those sugars are called trioses. There are those that have 4 carbon, and those sugars are called tetroses. There are those that have 5 carbon. Those sugars are called pentoses. There are those that have 6 carbon. Those sugars are called hexoses. There are those with 7 carbon. Those sugars are called heptoses. Now, the word pentose, you know what, by saying pentose sugar, now you know what type of sugar is. This sugar belongs to pentoses, meaning a 5 carbon sugar. In 5 carbon sugar, we have a different type of sugars. We have the ribose, as you've seen here. We have ribulose, we have xylose, we have xylulose, we have lysose. You will see quite a number of them when we come to that. So here, what we have is a pentose sugar, and that pentose sugar specifically is ribose. The other one is the oxyribose. I will shortly explain the ribose and the oxyribose. It is ribose that undergo a reduction to form the oxyribose. So when we are talking about the nucleotide contains a pentose sugar, and this pentose sugar is ribose and the oxyribose. And we know that the oxyribose is formed from ribose, which I'll explain shortly. We also have a phosphate group. This phosphate group is nothing but an oxide of phosphate or an oxide of phosphorus. Esther, please unmute yourself, Esther. You are distracting us. Esther, did you hear me? So it can be 1 phosphate called monophosphate. It can be 2 phosphate called diphosphate. It can be 3 phosphate called triphosphate. And we'll see all the situations where you can have 1, where we can have a 2, where we can have a 3. Now, starting with nitrogenous base. The nitrogenous base sorry, I think I will move to this one so that I will come back to the other slide for easy understanding. Now, the nitrogenous base. Now, I mentioned that the nitrogenous base, they are grouped into 2 parent groups. We have the purine and pyrimidines. These are parent compounds, purines. And you can see the structure by the left hand, top left-hand side. Did you see it? Did you see my puzzle?
Yes, sir.
Yes, this structure is the purine structure. It is the parent molecule here. And when you look at this structure, you will see that this is a 9-member ring structure. It has what we call pyrimidine ring, which is this aromatic ring. And then attached to the aromatic ring, we have this, which is called the imidazole ring. So pyrimidine ring fused to imidazole ring to form purine. And this purine, the atom in the purine is numbered in anti-clockwise manner. You can see nitrogen here is numbered number 1. It goes in anti-clockwise. We have the carbon 2. We have nitrogen 3. We have carbon 4 like this. Are we together? Are we together? Now this purine have 2 children, 2 sons. Like I said, this is the parent molecule. The first one is adenine. The other one is guanine. When you look at the structure of this molecule, now, how do we have adenine? Adenine is just an amino group attached to carbon 6 of this molecule. So it is 6-aminopurine, meaning here this is carbon 6. Are you seeing? Class, are you seeing?
Yes, we are seeing, sir.
This is carbon 6. So we have 6-aminopurine, meaning an amino group is attached to this carbon. When an amino group is attached to furin, it become what? It become adenine. And we can see adenine here. And on carbon 6, we can see an amino group attached to it. Can you see adenine and the amino group?
Yes, I can see.
You can see my cursor on that, yes. So, in the case of guanine, from the parent molecule, when you look at the parent molecule, we say guanine is what? 2-amino, 6-oxypurine. 2-amino mean an amino group is attached to the atom 2, which is carbon. Here, carbon 2, you have an amino group attached to it. Then 6-oxy, meaning this carbon is double bonded to oxygen. Of course, when you have a double bonded to oxygen, this double bond here between carbon and the nitrogen will disappear because the available electron must have been taken up by oxygen. And we know oxygen is a highly molecule that require electron. So it will take up the electron here. So, and we know that availability of electrons, it will bring about the double bond here. So once you have a highly electronegative molecule that can easily, electron, that can easily take off electron, it will take off the electron. Now you have this carbon double bonded to oxygen. And once you have this carbon double bonded to oxygen, and you have an amino group attached to carbon 2, we have what we call guanine. When you come down to this structure, you will see at carbon 2, we have the amino group, and at carbon 6, you have an oxy group. Class, are we together?
Yes, sir.
Yes, sir.
Yes. So this is some unusual purine bases. I think I intentionally, I wanted to remove them because I don't think they are important for this class. So I think you can just ignore these ones. So for pyrimidines, what happened? In pyrimidines, we said this is the parent molecule. Pyrimidine is the parent molecule. And pyrimidine have 3 children. Unlike purine that have 2 children, pyrimidine have 3 children. So when you look at the parent structure, pyrimidine have a 6-member atoms. So just like an aromatic ring, it has a 6-numbered atom, and these atoms are numbered in clockwise direction. Also, different from purines where they are numbered, the atoms are numbered in anti-clockwise. Now you can see nitrogen 1, nitrogen 1, carbon 2, nitrogen 3, carbon 4, like this in a clockwise manner. So you have these atoms numbered in clockwise manner, and we said it has the 3 children. These children are cytosine, uracil, and thiamine. Now, what is this cytosine? Cytosine is 2-oxy, 4-amino pyrimidine, meaning 2-oxy, this on carbon 2, it is double bonded to oxygen. 4-amino, on carbon 4, it is aminated or attached to an amino group. Now you can see the cytosine here, an oxy group and an amino group, while uracil is 2,6-dioxy, meaning a carbon or atom 2, which is carbon, and atom 6,